helped me and others through this is the general level of interest, intellect, and just engagement from all members of the population. And talks like this are so important to myself, because what you read about in the newspaper and what you hear on the news, sometimes it's nice to have a bit of context, or hopefully something from this that you can go back to and say, oh, I understand how that particular sentence or that thing that came out of somebody's mouth came about. So that's what I'm going to try and do over the next probably about 35 minutes or so, and then hopefully leave plenty of time at the end in case there's some questions. What I'm going to try and do is talk about these variants that you hear so much about in the news. I'm going to try and explain how they arise, and I'm going to try and explain why we need to worry about them, but how we need to also sort of temper that worry with being able to get back as much as normal to the world that we had before coronavirus came out, and how that may look as we go forward. I'm not going to, I'm going to show you real data, as I hope you're expecting, but I'm going to try and guide you through how we get to that data so that it comes alive, rather than just descending into jargon. I hope we'll see at the end how successful I've been. So I just wanted to start off by outlining the enemy, if you like, the virus. This is a picture of the virus, and I'm looking at another screen here. If anyone can still see me on screen and I'm looking away, it's because I'm looking at a big screen where I can see the slides. But this is the virus. It's one of a family of viruses. There are the coronaviruses, based on how they look when you look at them down a microscope. They're tiny as with any other virus. This is exploded massively for the photograph, but you could fit about 1,000 of these across a human hair, going from side to side. And something that was calculated recently was that if you collected all of the virus in the world at the moment that was infecting people and crushed it all down, got rid of all of the space around it, you could pretty much fit it all in a standard can of Coca-Cola, and that gives you an idea of the scale of the virus relative to the amount of disease it's causing. When we zoom in, we actually see what the virus looks like, and it's a typical virus in that packed inside it is its genome that it needs for being able to encode its 14 proteins, and I'll come back to that in a moment. It wraps itself in a bit of lipid that it gets as it goes out of a cell, and then it studs into this all of these things called the spike protein. You can just about see them here. And all the virus really is about is taking its genome and injecting it into a cell, replicating it, and making sure that all of its individual genes in the virus are expressed and form a new virus particle. And really and truly, it's all about translating the genetic code into the proteins that the virus makes. And this is the one that we focus almost exclusively on in the world of treatments and vaccines, or particularly vaccines at the moment. It's called the spike. It sits in the envelope of the virus, and it's this bit at the top here that the virus uses to engage with the cells that it infects so that the virus can latch on and then get inside the cell. And this particular protein is made up of 3 identical units. And where you can see the bobbly bits on the edge, that each one of those represents an amino acid. And amino acids are just part and parcel of what we all have, building our own proteins. And all the virus tries to do is plunder the treasure trove of the cells that make up our body, using all of our resources to replicate its genome, make new virus particles and new virus proteins. And really and truly, most of this talk is going to focus on amino acids. And each one of these amino acids that joins together to make up the whole protein chain is part of 20 that we have as a human being that we use to make all of our different proteins. Some of them we can synthesize from within our cells. Some of them, essential amino acids, have to come from our diet. So for instance, tryptophan, you find a lot of tryptophan in protein-based foods and dairy proteins, so chocolate, oats, dates, etc., milk. And within this protein of the virus, there are many tryptophans that go together to make up the virus spike protein. So what we're going to talk about for the next 30 minutes is how this thing and how this genome changes and what that does for the virus and how we then go on to track it around the world and what it means. So just to give you a few more facts around the virus, if you were to go from one end of the genome all the way around to the other, there would be 30,000 of the building blocks called nucleotides. And for every 3 of those that come together, you make 1 amino acid. So in total, there's the potential to make about 10,000 amino acid, encode for 10,000 amino acids. To put it into scale, this genome is about 100,000 times smaller than the human genome that is in every single cell of our body. And whereas our human genome encodes in the order of 200,000 proteins, as I said, this virus only encodes for 14. And it's only those 14 proteins that the virus needs to make itself, transmit on, and cause disease. All viruses have a real problem, though. They strip themselves down and they don't have an awful lot of baggage for all of the things that we have as if you like, a higher organism. We want to maintain the integrity of our genomes as much as possible because mistakes cause proteins to break. Viruses generally tend not to do this very well, and this particular virus, every time it makes a copy of its genome, will introduce about 1 or 2 errors. So 1 or 2 errors in every 30,000 of these nucleotides. And as the virus passes from one person to another, then the virus species as a whole, all of the viruses, accumulate about 2 of these per month. But that really means when we've got hundreds of thousands of infections every day around the world, the virus is effectively exploring all of the different combinations of individual mutations that it can make throughout its genome as it passes from one person to another. So variants are just a natural thing that viruses do. We just get to see them, particularly at the moment when we're looking so hard at the virus, and we need to understand their consequences. So what are these? So I've mentioned things like the genome of the virus, the protein as it all comes together, and the amino acids that go together to make up this protein as they form long chains. How does this all link together to give us the variants? Well, in a way, all of these proteins, all of these amino acids, when they are made in the cell and turned into a protein, will daisy chain themselves together. And you can follow this daisy chain by when we use X-ray crystallography to solve what these proteins, even though they're tiny, incredibly small to see, you can work out their exact 3-dimensional structure using X-ray crystallography and the very things that the UK was one of the pioneers of developing a number of years ago now. And you can just about follow the protein, you can follow the chains. It gets all very confusing as it gets together and it looks like a bit of a bowl of spaghetti, but nonetheless, all of these chains are made up by joining the amino acids at this end and daisy chaining them together. And then the functional bits of the amino acid that make each amino acid different hang off this chain and then enable the protein to fold up and give the protein its function. The way that the virus makes this protein in the end is to start off with its genome that's made up of these 4 nucleotides. And every time you get 3 of them together, they are the genetic code for a particular amino acid. So that means as you roll along the genome, working out which amino acid to put in, if you come across CCU, a C, a C, and a U, then the amino acid you encode is a proline. And if the next one along is an AAC, the next 3 in the chain, then you code for an asparagine, and you join those 2 together, and you've got a little bit of a protein. And then you keep doing that, rolling all the way around until you've made the whole protein and it folds up in the cell and you start making a virus particle. So variants, and this is all they are, is when the virus as it copies its genome makes a mistake. And so rather than having a CCU, it now has a CCC. Now, in this case, that doesn't mean that you change the amino acid because this code is redundant and there are different ways of getting the same amino acid. So that gives you a bit of tolerance in the genome for mutations. But sometimes you can make a mutation and change the amino acid. And this particular change here of an A to a U in one place in the virus genome is one of those changes that gave rise to the Kent variant, or the variant that arose in Kent or was first discovered in Kent, that's caused us so much trouble over the last, well, really about 4 or 5 months. Just that one single change in the genome. To be fair, there were a number of other changes in the genome as well, but this one seems to be one of the most important one, and it encodes for a change just at the tip here of this protein where it interacts with the cell. So that's the background, that's the substrate, if you like, for everything that we're going to just talk about. Copying the genome, making errors, those errors not being silenced, but encoding for a change of an amino acid, and the amino acid ending up in a place in the protein where it's important for its function. So how on earth do we go about finding these things? Because, as I say, this is tiny and really quite long for a virus. Well, as Eric mentioned, for a number of years now, many labs in the world, including many in the UK, have been setting up systems for being able to do this. And we reasoned very early on that if you had an outbreak and people were getting swabs done from their nose or the back of their throat and sending those swabs on to be diagnosed in a hospital lab or a diagnostic lab as to whether you're infected or not, using the PCR test that we've known to come and love or hate depending on whether it comes back positive or not, then we reasoned that once you've taken that sample, if we could just divert 5 or 10% of them after they've been tested for the presence of the virus, amplify all of the virus up in the laboratory, throw it onto these modern DNA sequencers, and then use computers to stick it all together again, we would get an idea of the virus genome in the person that had a positive test. And from that, we would be able to determine if there are any changes in the virus genome from one person to the next person to the next person, and start to track these so-called variants. And we're one of the world leaders, if not the world leader in this at the moment. COG UK has produced just shy of 500,000 genomes today, and it's done more than half of the world's genomes so far. The rest of the world is now catching up, which is fantastic, and that's what we need to keep doing. But these are our substrates for following these variants as they go around the world, trying to track those single mutations or collections of mutations that make up a variant. So how does this all hang together? How do these variants come about? Well, this is a schematic that was just taken from something that was recently published. If you imagine here, this is the person that became infected, and they're infected by a small number of virus particles to start with, and say the one that's the red variant gets going in that person to start with. Over time, that virus will replicate in the person, and it will start making errors in its genome, and they will accumulate as different viruses represented by the different colors here within a person. Now, if that person is to transmit on the virus, at that particular moment in time, it's a random sample of the types of variants that the person has. And without too much surprise, the more common variant, in this case the red one, is more likely to transmit on. And if it does so, then it sets up a new infection, and then that new infection will grow and multiply within that person, and you'll get more variants, and they'll become colored, and that chain of transmission will go onwards. Just occasionally, the rarer variant will transmit. This is one that has some of these mutations in the genome, and that will infect a new person, and then it will start to grow. And now you have two lineages or two variants of the virus around, the red one going down this way and the blue one going down this way. And if this person keeps being persistently infected for a while, it takes them a time to recover, then perhaps they start to transmit on a collection of viruses to the third person they infect. And again, the process starts. And this is happening every time a new person's infected. Variants arise, transmit on with various probabilities. And the things that help the virus on are whether the virus has an advantage in a person to become the dominant red one or a dominant blue one, whether there are blocks to the virus being able to transmit on. And in the end, the variants that win out are those that have an advantage. Some are better than others, or they're lucky. They happen to be in a group of people perhaps here, that all went off to a particular house party at a particular moment in time, and this person goes from 1 to 20 people infected, whereas this person down here got their PCR test and stayed at home, and that lineage dies out. So you also need to be, so being fit and lucky are the two things you need to do. What you'll start to notice is that this looks a bit like a tree. This person goes out and infects them and them and them. And we can use all of the mutations in the virus to piece together this tree of transmission, particularly because we know when the people are becoming infected and giving their samples. And so you build these family trees. As person 1 transmits to person 2, transmits to person 3, the variants accumulate mutations in their genome, and these act as markers. And depending on how we count them, we can work out both how distant, how many transmissions have happened for this virus to become this different, and normalize it or make it sort of interpretable by knowing that this sample was taken before this sample and this sample was taken before this sample. So we effectively build a virus family tree based on the variants that the virus has and the times of the sample was taken. Now, when you piece that all together, this looks all nice and simple, and you can see the family tree, and it might be the family tree of the transmission of someone that's transmitted onto 3 people. When you start to accrue that across the world or the country, then you start to have trees that look like this. So this is a collection up to the end of April. This isn't all the genomes in the world because there's just shy of 1 million genomes now, I think. No, probably about 750,000. This is a sampling of them, and every single dot on here is a new virus genome that's been sequenced. And what you can probably make out from this is that there's a fairly strong trunk to this tree. There's a good backbone, and then there's loads of branches coming off. And some branches are populated all the way down by virus leaves, and some branches are very, very long and then suddenly explode at the end here. And these things tell us a lot about what's happening to the virus over time as it spreads around the world. And you can see here all of the countries of the world pretty much have many, many different variants all the way around. And when people talk about variants, what they're talking about is picking 1 or 2 of these that seem to have become dominant, either as a group of branches that are all related, or because they're all taking off in one part of the world. And that gives us an idea that these variants are important. Now, the thing that starts to stick out for people that spend their life looking at these trees is that when you see a branch like this, lots of internal examples, what you're looking at is the spread of the virus from one person to another, to another, to another, gradually changing over time all the way through in a nice clock-like way. We know what the virus has been doing. We've got examples of it over a long period of time. But what seemed to happen towards the end of last year was that we started to notice these very, very long branches where we had no examples of the virus from anywhere else in the world. There's one here, and there was another one down here. These long branch lengths. What they effectively say is that there's a lot of missing evolution. The last virus we saw was round about here in April, then it disappeared, and it reappeared down here with a huge range of mutations. The length of the branch is how many mutations the virus has accumulated. And this group up here is actually the Kent variant that occurred in Kent, then spread throughout the UK and then the rest of the world. As you can see, it's now all through the world. So these long branch lengths tell us something about the nature of the virus as it transmits along. It's accumulated a lot of genetic change. We don't really know where it's accumulated that change. So it's all very well gazing at these viruses and saying, oh goodness, there's a lot of change. How do we go from picking, say, this blue one here and working out whether that one is more interested and important to us than these ones up here? Well, unfortunately, you can't do this only from the looking at the genomes of the virus and tracking the variants. You have to bring in other biology, and you have to start to create layers of information. The sort of things we look for, I'm just going to exemplify on the next couple of slides. We knew many, many months ago now, right at the beginning of the epidemic in February of 2020, we had a blue collection of viruses. These are the ones that seem to come out of China. And they all had a particular change, again in the spike protein. And when you focus in, and here's the backbone, and here's the amino acid, and here's this number amino acid, it was amino acid number 614. And this amino acid changed from a D to a G, round about here. And that seemed to give the virus an advantage, because from that moment on, this virus dominated. It was fitter, it was faster, and over the period of about 6 months, it swept around the world and completely replaced the blue version, the D version. So this is what we call a variant that sweeps to fixation. Basically, the genetic mutation sweeps through the population, and then pretty much now in the world, there only are G versions of this virus at this particular amino acid. A lot of work was done on this, and we found out that this particular amino acid grabs on over here slightly harder when it's a G than when it's a D. This means that this protein is slightly more stable, and that means the virus has a slight fitness advantage in that it replicates slightly better and transmits on to new people slightly more than the blue version. And you only need small differences in these viruses for them to gain a fitness advantage and sweep through the population, transmitting on. It becomes a straightforward foot race. The fittest, fastest virus wins out. And this was an example of its sweeping to fixation and new mechanisms that we understand. So what did the so-called Kent variant look like? Well, I made this just this afternoon to see what the Kent variant looks like in the world. The Kent variant has a particular defining mutation here at amino acid 501 in the spike protein, the one we keep looking at. And this one is right at this tip here, exactly where the protein interacts with the cell receptor. And the cell receptor is in green up here. And what this amino acid change seems to do is that it makes the virus stick slightly better to the cell receptor, and therefore it has an advantage in that it is able to it seems to infect cells slightly more efficiently. And what we've seen since it was first discovered, round about since it first arose, round about the late summer, early autumn, and when it was first seen in the UK, when we first started to detect it, is this massive accumulation of viruses that have this similar mutation throughout the world, suggesting that this is also a virus that is fitter and spreads throughout the world. Now, we were very good in the UK about being able to see this happening and try and work out what on earth it meant. And an awful lot of work done very quickly in December last year and over the Christmas period by people, fortunately not myself, but across a number of people, particularly in London, Glasgow, Warwick, and other places in the UK, came up with these sort of data. The reason we noticed this variant is because it had unusual properties. In the southeast of England in November and December, even though they were under tiered restrictions, we couldn't keep the infection under control. And when we first noticed that the genetic variant, this so-called B.1.1.7 Kent variant, was there, it became very clear that it was also linked to a failure of part of the diagnostic system that was used to detect the virus in all of the samples that we were collecting from people. So now there was a method of tracking this virus, even though we didn't sequence all of the genomes, this so-called S gene dropout or S gene target failure. And it meant that we could go back retrospectively through all of the data that was accumulated through the diagnostic PCR, the diagnostic testing of the population, and ask certain questions. And it became immediately clear that there were no underlying epidemiology of this virus that would explain why it took off so well in the human population. And what you could see, particularly in Kent, and also then in London, and then as it moved through the country, was that when you had periods of lockdown, and this is the lockdown through November, even though the non-Kent variant was pretty much under control, this lockdown didn't seem to do very much at all to the Kent variant. And when we came out of lockdown, then it took off. And this is how we ended up with the epidemic across the UK from December through to pretty much where we are now. So this variant had a fitness, And this one was growing, even though this one wasn't. So whatever was going on here, this particular virus had an advantage. And then just slightly behind Kent and Midway, where it particularly seems to have arisen, then the same pattern happened in Oxford and Buckinghamshire and London, and then eventually just a bit later on, now in January, Stoke-on-Trent. And we have these for across the whole country, such that we can see that over time, if we count how much of this variant is present in different places in the country, it started off in the east of England, in the southeast, and over a period of time from the beginning of November to pretty much February, this virus became completely dominant in all of the cases, and then gradually moved through the country, becoming dominant through all places, such that in the end, even all through the country, the variant has led to the dominance. And now we can say without any concern at all that this virus is a dominant fit virus, and unfortunately it is also spreading throughout the world. And this is the evidence that we need, both the genetics and then some real world epidemiology and some mechanism how this virus becomes fitter to see what's to enable us to call it a variant of concern. So if we were to piece this all together, because there's been so much looking at this throughout the world, I'll bring you back to this spike protein, and here it is again, and here it is sticking onto the receptor that gets the virus into the cell. And this is a sort of an exploded view of it. Every red dot on here is a variant that is present in multiple different viruses around the world. They're not all linked on the same virus, so some of them just occur as one-offs, some of them are pieced together in these so-called variants of concern, and you'll notice that there's particular regions of the protein that seem to harbor these. There's this region here called the N-terminal domain, there's a region here called S2, and then there's the bit up here where the virus binds to the receptor and causes a load of mutations in here, and you can see them here, and if you spin it round and look in through the backside, you can see these here as well. And largely the virus is doing two different things. It's optimizing itself to spread efficiently in humans, because originally it wasn't spreading in humans, it was whichever animal reservoir it was really in, and so the virus is fine-tuning itself to spread more efficiently. And that's some of the ones we've seen, perhaps that D614G and the Kent variant. But we also know that these sites are the very same sites on the virus where when you make an immune response, either because you've become infected or because you've had a vaccine, these are the sites where the antibodies from your body latch onto the virus to prevent it infecting cells. So by the virus optimizing its ability to spread, it is also probably optimizing its ability to start to escape some of the immunity that has been generated either from an infection or potentially is there to block the virus as a vaccine. And this is why we're so concerned about these variants of concern, because of the double edge that they have of being able to be fitter viruses, but also because they might start to escape some of the immunity that we're generating in the population. And that's why these have risen to concern. And that's just what I want to spend the next, the final 10 minutes just talking through what we think this may mean. So if you're interested in tracking these, you can find these all over the world, all over the different agencies of the world. The Centre for Disease Control in America runs a similar list. The WHO runs a list, and the UK government publishes I think monthly, the variants that we're tracking. And we are currently in the UK tracking 4 variants of concern. This is the one at the top I've been talking about, the so-called Kent variant, but there's also the South Africa variant, a Brazil variant, of which there's 2. These are all variants of concern, although this one is not, sorry, this is a variant of interest. So a variant under investigation, which are all these VUIs, becomes a variant of concern when we know that it either transmits more efficiently or it has an escape from some of the immunity that you generate as a consequence of infection or vaccination. And if it transmits more efficiently and has immune escape, then it becomes a variant of concern for sure. And the South African one is the leading candidate here. And all of these VUIs, these so-called variants under investigation, are the topics of intense sampling and research, such that we can build up the evidence that either says we don't need to be concerned about them because they've arisen and we don't see them anymore, or we do need to be concerned about them because they've arisen, we see more of them, and we know that they're escaping some form of immunity. So that's how we make the distinction between the variants of concern and the variants under investigation. So what about this immunity? Now, I just want to spend, I know you've had a vaccine talk before, and I'm not going to go into all of the vaccines that we're deploying around the world or in the UK, but I just want to talk briefly on what's known about what's called vaccine efficacy. Now, the way we work out how well a vaccine works is to do a controlled trial in the population, and it could well be that some of you that have been listening now have been part of some of the vaccine trials. One of them that was conducted by a vaccine that's being deployed in the UK is this Pfizer-BioNTech vaccine. And what Pfizer-BioNTech did was to get 43,500 people together, split them into those that got the vaccine and those that didn't, and then follow them over time for how many of them, as a percentage, ended up with symptomatic COVID. And that could be seriously ill, or it could be mildly ill, but something that was recognizable by symptoms. And when you follow this over time, so here's days along here, and here's the increasing frequency of people that get symptomatic disease, you can see very clearly those that didn't get the vaccine, over a period of 112 days, out of the 21,000 people, round about 200 or 300 ended up getting disease, about 2%, or whatever the number is that gives 2%. Whereas those that had the vaccine, after initial point to get the vaccine working, then almost nobody went on to get disease after that. And the difference between these two is the difference of how effective the vaccine is, or the vaccine efficacy. And you end up seeing quoted lots of numbers, 95% vaccine efficacy. In fact, all of the vaccines around the world that have been tested so far have almost 70% efficacy or more. What on earth does that number mean? People tend to think that means that 95% of people will no longer get the infection. Unfortunately, it doesn't mean that at all, because what this number is a measure of your relative risk of having the infection if you have the vaccine. And so that means you've got a decrease of 95% in your relative risk. Now that might sound complex, but it's actually really quite simple to follow if we think from these numbers. So this was a vaccine trial made. 2% of the people that didn't have the vaccine got disease. 0.1% of the people that had the vaccine got disease. So there's a difference between the two. There's a difference in relative risk. So all we do is to take those two numbers. 2% in the placebo arm got disease, 0.1% in the vaccine arm got disease, and we calculate the vaccine efficacy. We'd like it to be a percentage in the end. So this is one way of doing it, and there are others, but so one way to do it is to make it a percentage in the end by multiplying by 100 and say that the vaccine efficacy is 1 minus the attack rate with the vaccine, that is the placebo group, divided by the attack rate, 1 minus the attack rate with the vaccine, this number, divided by the attack rate with the placebo, this number. And so if you plug these into the equation, 100 times 1 minus 0.1 divided by 2. That is 100 times 1 minus 0.5, if you can do it in your head or you do the calculator, which is 100 times 0.95, which is 95%. So the vaccine efficacy, your relative risk, is reduced by 95%. So it doesn't mean that 95% of people are protected from disease. It means that a person is 95% less likely to become infected relative to the current chance of becoming infected if you didn't have the vaccine. But this has a nasty kicker in it, because you can get 95% vaccine efficacy by having 10 times more disease around. So if we were in a condition where actually you had 20% of people in the placebo group became infected, and 1% of people in the vaccine group became infected, you can plug those numbers in. They're 10 times larger. You've got 10 times more infection around, but the vaccine is still 95% efficacious. But now you've got 10 times more people with disease. And what that really means is that vaccines work best when there is less disease around, and when they are able to exert their maximum their efficacy on a small number of infections. So to successfully exit lockdown, we've got to keep the disease under control. We've got to vaccinate lots of people quickly to get this 95% of the population level, and we've got to keep the amount of infection low and avoid variants coming in. Because if a variant nibbles away at this efficacy and starts to decrease it a bit, then this number is the one that goes up, as well as relative to this number. That is, the vaccine will protect less people given the rate of infection in the community. So how can we measure that? So the last couple of slides. We really don't know how to equate the vaccine efficacy that we measure in clinical trials that I've just described, that 95% reduction in your relative risk, to something that we can measure from a sample obtained from somebody that has a vaccine. We have an idea, but we don't have any formal proof yet that we can do that. But what we think we can do is just measure the amount of antibodies that are in the blood of a person that's had a vaccine or has had the infection. And if your blood is packed full of vaccines, you can dilute them out many, many times, but you've still got a potent cocktail of vaccines, of antibodies, even though you've diluted them quite a long way. So here's a 1 in 100 dilution, and then 1 in 1,000, and 1 in 10,000. Somebody that has no antibodies or a very low amount, once you start diluting them, then by the time you get down here, there's no antibodies left. And we can test how many antibodies there are in somebody's blood sample by just adding a standard amount of the virus, either with no antibodies present at all, so that's our 100%, or when we've got these dilutions. So we add the virus into these dilutions, or here is our control with no antibodies, and then we plate the virus onto cells, and let's see how many viruses can infect the cell monolayer. And the virus, each one of these plaques that's here is where the virus has infected the cells and killed all the cells around it. So these are called plaques. And when you've got antibodies there, the antibodies grab hold of the virus and reduce the number of plaques, such that when you've got a huge number of antibodies, you have either only 1 or 2 plaques or no plaques at all. And in order to compare between people, we say, well, what is the dilution of somebody's blood that takes this 100% and reduces it to 50%? We call this the 50% plaque reduction. So the dilution of somebody's sera that leads to a 50% reduction in a standard amount of virus that's put into the experiment. The way this looks is now you can compare to different viruses. I should say it doesn't matter whether you put the original virus in here or whether you put the Kent variant or the South African variant or the Brazil variant. Each one of these will give you a readout of how your antibodies inhibit the virus from making these plaques. And so this has been done, and this is the penultimate slide, and then we'll stop for some questions. And this is the dilution, and here are the different viruses along here, and every dot here is a person that's donated some blood, and it's the same person's blood tested against the original virus, the Kent variant, then the Brazil variant, and the South Africa variant. And what you notice here, and these are people after they've had an infection and their antibodies are present in their blood, that in general, as you go from the
Paul, I think we've lost you. I think we've lost you at your sound. You've suddenly gone on mute. Can you unmute yourself, or Rachel? Can you unmute Paul?
You're absolutely right, Eric. How on earth did I do that? I beg your pardon.
I'm so sorry. It was my fault because I was trying to mute whoever was unmuted, and then I made a mistake. I'm really sorry.
That's all right. That's okay.
Carry on.
Okay. There's only one more slide to go, so I'll just crack on. Can you see them again?
Yeah, the slides are perfect, Paul. Can you just run over the last little bit of the graph on the left?
That's right. So so what it is each one of these dots is a sample of the blood from a patient, and then they are assayed to see how many of the antibodies in the blood can give an inhibition of the virus that was originally came out of China. So these are the values here, and then test the same blood sample against the Kent virus, the Brazil virus, and the South Africa virus. And then you can track how an individual's sera, blood, responds to the different viruses. And in general, the Kent and the Brazil virus have about a 2 to 3-fold decrease. You can see the lines going down. The virus is slightly harder to inhibit with the anti-sera, the antibodies from these patients. But the one that causes us a worry is the Brazil variant. This one, if you've previously had an infection with this virus, many people don't mount a very good response at all to this South Africa virus. And that's about a 10-fold decrease. And we see the same sort of thing in people that have been vaccinated. So here's the Pfizer-BioNTech vaccine. It protects well against the virus that came out of China, protects well against Kent, protects fairly well against Brazil, but is starting to come off the boil a bit against South Africa. Now, we really don't know what this means, and this is the holy grail now of what we're all trying to discover, because it probably means something, and it probably means something not very good, but it's definitely not a complete disaster for the vaccination program. And so this last slide just says exactly this. If we measure the antibodies in somebody's blood, and then try and correlate that with how well the vaccines work in their efficacy, their relative risk of getting symptomatic disease, there's a trend. The more antibodies you have in your blood, the more potent the vaccine is. The less antibodies you have in your blood, then the weaker potency. Now, all of our vaccines that we've got at the moment are up in this greater than 70%. Here's the AstraZeneca, here's the coronavirus from BioNTech, Pfizer, other ones that we're about to deploy in the UK. They're all up here. But the worrying thing is, if we have a 10-fold decrease, then something that is up here protecting from symptomatic disease in 90% might come down to only protecting 50%. And when we go out into the field and we look at clinical trials for the J&J, the AstraZeneca, or another vaccine called Novavax, we see that against the original virus, we've got these good efficacies, 60 and 90% against clinical disease, and really incredible efficacies against severe hospitalizable disease and death. But the worry is that when we see the variants in some of the vaccines, we see this decrease that fits exactly on this curve. So if we take Novavax, that's up here, round about 90% efficacy. We think that this causes a 10-fold drop. So if we come down here, which is about 2, and go down to 10-fold less, it's about here, and it gives us an efficacy somewhere in the 50 to 60% range. And when we look in the clinical trial in Africa, it is in that 50 to 60% range. So we're worried now that these variants will mean that we still have symptomatic disease and spread in the population, but we hope and think that they may still prevent severe disease, and therefore filling up of the hospitals hopefully will not come about because of the variants in such a bad way as if we didn't have the vaccines at all. But nonetheless, the virus will still be able to spread and transmit around. And the concern is that eventually it will pick up enough mutations where the efficacy will drop quite a bit. So I'm just going to leave it there. I'm not going to bother running through these, but what I just want to emphasize is that there's not a lot we can do about this because the virus is a pathogen and variation is natural and we can track it. We can update vaccines, and the UK will be deciding whether it needs to revaccinate with to boost everybody's immunity or to vaccinate with a variant later on this year. And when it comes to things like influenza, we can keep ahead of the variants, and that's why we update our vaccines whenever they're needed, and it can be every year. And we think that there's a chance that we can do the same with the coronavirus if we need to. Our biggest problem now is how we roll out vaccines to the whole of the world very quickly to stop the amount of transmission that happens and the accumulation of new variants in other places. So we're not out of the woods. We have a lot of tools at our disposal. We know we have an awful lot of knowledge. This amount of knowledge would normally take about 20 years to accumulate, and it's been accumulated in about a year. Everybody's tired of wanting to live under the regimes that we have to, but the urge is for people to be gentle as we come out of lockdown, get your vaccines, have both doses, and still maintain some element of social distancing as we come out of lockdown, such that we can try and keep the UK at least protected as we can from the variants that are circulating. So I'm just going to leave it there. And I know we're close to 8:30, but I don't want to keep people. I'm happy to stay on later than 8:30 if there's questions. And I forgot to put up the advertisement for your next talk, which is on the 19th of May. And if I'm still sharing my screen, I'll just put that up now so you can just see. Hopefully that's back up.
Paul, thank you very much for an absolutely fabulous talk. You've taken the broad sweep of virology, infectious disease, molecular biology, and you've taken us from the genome, and you've dug us all the way down to the interface between the virus and the cell. What a journey. Thank you very much for that. I think I feel quite sure that we've all got a much better understanding of the significance of these variants, how they fit into the general picture of how the virus approaches the cell, evades antibodies, etc., etc. So thank you for that tour. That was wonderful. And I'm going to open everything up for questions now.
I can see one question in the chat whilst people are coming off microphone, if there are questions, from Romy, who asked, scientifically, what's the difference between a mutation and a variant? Well, you can't have a variant without a mutation. The mutation is the fine description of that mutation. What we tend to think about are variants. The variant is something that then exists as a viable virus that we can see, that we can grow, rather than a collection of mutations. But in a way, they are interchangeable. And so the terminology that's come about for the SARS coronavirus is to talk about variants rather than to talk about mutants. But they are, in a sense, interchangeable.
Great. Well, how about the question, Paul, about do you think from Ying Lu, do you think the mRNA vaccine could be hopeful to catch up with new potential variants? And maybe I have my own question there about, can you predict what variants are going to come along? Do you have a contingency plan? If you were going to be said to design the catch-up vaccine in the autumn, what would you go for?
Thanks for that one, Eric. That's very kind of you. So to Yinglu first, yes, the mRNA vaccines are a revolution to this. We've never had them before, and there is the potential to update them very quickly. And indeed, in the US, the two vaccine manufacturers, Moderna and Pfizer, are doing vaccine updates, and they're updating to the South Africa variant. And they are starting clinical trials right now as we speak. And the regulatory bodies know what sort of data they would like to see in order to be able to give a license to such a variant update vaccine. And in the UK, we're doing the same thing, both what we did with the vaccine task force before, which is to talk to the manufacturers to forward procure variant vaccines, and also to have our own homegrown version of an RNA vaccine that will be produced where we look after our own supply chain, with the aim to be ready for the beginning of 2022. So very quickly this is all happening, and we wouldn't have been able to do it so quickly before. For your question, Eric, can you predict the future? Probably you can have a reasonable guess, hence the reason people are going for the South Africa variant. But actually, in reality, it's really difficult, because if we have another wave in the UK, it will reflect pretty much what we receive into the UK from the rest of the world as the dominant form of the virus that will take off. We might get a resurgence of things that are homegrown, but in our past two waves, most of our epidemic has started off, at least, from what we import, the Kent variant perhaps being the exception to that. There is an awful lot of work going on to try and forward predict, and to try and, if you like, throw some vaccines ahead of where the virus might go in order to block off its avenues. But this has never been done before, and therefore it's a bit experimental. But I'm pretty optimistic that perhaps not this year, but over the next year or so, we will learn so much more about how you can get ahead of virus evolution and vaccinate intelligently in a forward predicted way, that for the future, I think I'm more optimistic. For the here and now, we know that the vaccines we have aren't completely dead by a long way. We know that we can boost, most likely with the existing vaccines, and we should be able to replace with a variant vaccine. And we know that if all else fails, we can bring things under control by going into a lockdown, but nobody wants to do that anymore.
I mean, I guess with flu, we've always had a harbinger of the future from the southern hemisphere. So what happens in the southern hemisphere winter is a good harbinger for what's going to happen to us in our northern hemisphere winter. And the seasonality aspect is not there with coronaviruses. Can you comment on the seasonality and
Yes, I think seasonality is something that has been often mooted, because the four existing human coronaviruses have a seasonality to them. But really and truly, seasonality, when you have an endemic virus, comes about through a complex mixture of virus transmission, the immunity in the population, and when you've got a lot of immunity around, it's harder for the virus to transmit, and perhaps you just need to all be together indoors a bit more. So the seasonality might be a consequence of how the virus ends up having to transmit. At the moment, this virus just has free reign. There's not that much immunity in the human population, and it really doesn't care that much about hot or cold temperatures, as we can see in India, in South America, in Texas. If you have lots of humans together in an enclosed space, the virus just happily transmits on.
Now, there's a question here from Claire Snells. It's a fairly long question, but it's really about A strains and B strains. So I think basically what Claire is saying is, we've gone from A to B, and we're now on P.
Yes.
Can we go back? Can previous strains that have disappeared become resurgent again?
So it's a great question, because this is one of the problems with flu, particularly with animal reservoirs of flu, that you can get things coming back. But actually, flu tends to move inexorably onwards, because it transmits happily in humans. At the moment, I think all bets are off, because what goes on in, say, Africa at the moment is different from in India, is different from in Brazil. And what may look like a resurgence in one place in the world, a reintroduction of, say, A into B, could be something brand new in another place of the world when they've not seen A before. And so what we've got are multiple plates spinning at the same time. And it's going to be very difficult, I think, to work out backwards and forwards movement of the viruses and how they take off. I think on the whole, though, what we're seeing is the virus getting better at transmitting. The the P variants from Brazil, the South Africa and the Kent variant, and probably the variant from India transmits on better, and it's hard to go back to a variant that transmits less well. So you might see some remnants of what was an old A variant, but it might be an updated form that transmits on better.
Peter Reynolds says, is there any evidence of protection produced in those who have been infected and survived?
Well, there is, in that, particularly when you look in some of the cohorts in the UK of healthcare workers, we see very well that the antibodies that they produce and the other side of the immune system, the T cells, are effective and productive, and we are assuming that that is what is protecting them from the decrease in disease that you see. Unfortunately, we know as well, it's just a facet of the immune system, is that your immune system wanes over time, your immune protection. And so someone that's nice and protected now because they had a good immune response to the infection, say in April 2020, it's now April 2021, and they won't be quite as robust. And the same applies to vaccines, which is why we need to boost. So it's a dynamic, but there is certainly evidence there. That's how we know the vaccines work. And that's how we know that some of the components of the immune system work so well. And I think the trick is to keep boosting it to a level where that protection is the dominant force and the waning of protection becomes a minor force.
Yes. A question from Matt Walters. Great talk. Yes. Are there any new promising antivirals in development that could be available soon? We had this announcement yesterday, a challenge by the government, and I think we've had a challenge from AstraZeneca as well. I think a number of companies putting money out there.
Yes, yes. So I'm not sure the cupboard's particularly full with novel antivirals. And if they are there, then they are going to need to go through the phase 1 and phase 2 safety, and that takes time before they go into an efficacy trial. What we've seen, and I'm sure you're as aware as I am, the recovery trial in the UK and similar things now springing up around the world have taken existing medicines such as dexamethasone and shown how potent it is in people that are sick. One of the drugs that is used in Japan for treating influenza seems to have some activity against the coronavirus as well. It's called favipiravir, not an easy name to trip off the tongue. And I believe that's starting to go into clinical trials now in various places in the world, including the UK. And because it's an antiviral, one of the problems that we find is that you have to get them quite early. And I think the announcement that came from the government was this magic pill that you'd have in your cupboard. And then if you happen to be around someone that tested positive, you'd pop this pill. Well, that is really what we're aiming for. But the reality is it's going to take a little while to get there. Favipiravir is probably the closest because we already know it's a relatively clean and safe drug. The problem is if you have it out in the general community for people to pop at the moment's notice, you've got to be extra careful with its safety. And we've seen recently how a 1 in 250,000 event for a vaccine is enough to really dent people's confidence. And that's an incredibly rare event. And with drugs, we've got to be aware of the safety profile. So safety first, but not a great deal yet.
Yeah, and I guess the other aspect of it is a bit like antimicrobials, antibacterials, is that if anybody ever did come up with a super antibiotic, the first thing is that the chief medical officer would lock it up and only use it in extreme cases. And of course, you do develop resistance to antiviral drugs as well. So there are problems there. Now, we're happily talking on here that there are still 45 of you out there who are obviously interested in the chat. Thank you very much for that. Just as we're chatting on, could I just say a big thank you to Rachel Unsworth, who is our event secretary. If this event seemed to go very smoothly and so on, it had nothing to do with me. It's Rachel and Paul's work, and thank you very much, Rachel, as well as to Paul for for a great talk. If many of you may not be members of the Leeds Philosophical and Literary Society, we have been going since 1819. We're in our 3rd century, and we are here to disseminate public understanding of science and the arts. And it looks like we're getting a global reach now with thanks to coronavirus. We've had people from New York and all over the world. If you're interested, please visit our website. We are going to be updating our website and making it even more exciting. And if you like what you're seeing, why not join us? We have a ridiculously small subscription fee, and you will get plenty of information from us, invitations, and we do produce publications which we distribute freely to our members. So that there are a lot of advantages in being part of us. So that's my little plug for LPLS.
I saw a question from Claire.
Yes. Yes, sure. Let's go back to Claire's question about the Uganda strain. What would you like to talk about that, Paul?
Yeah, so Claire points out quite rightly that these the A strains, if you like, in Africa seem to be accumulating very similar mutations, becoming the same sort of variants as the successful B lineages. And does this tell us anything? And I think this very much relates back to Eric's question of how can you predict the future? Well, I think it really does tell us something, and it's a property called convergent evolution, which is if something's a good idea and it has a selective advantage, the chances are it will occur more than once in a population of organisms that can mutate. And that is really our best chance of forward predicting where the virus is going to go. As at the moment, the virus is, if you like, wandering around in space, trying to work out what it needs to accumulate. And then once it's got its good collection of mutations, accelerate through the population. Because we're sequencing it and we can see all of its variations on a theme, we can try and put together and spot those that are common. And that's exactly what is going on. And that's how some of the more forward-looking vaccines are trying to be put together. The danger is that we're not as smart as we think we are, which is almost definitely true, and that we end up being fooled by what the virus is trying to tell us because we don't really understand why it's doing it. And it's nothing to do with vaccines, it's to do with something else. And so I think we've just got to be have a bit of humility and try and do the work as quickly as possible to understand how the genetics relates to things that we can measure and how they then relate to what we can do with vaccines and other therapies.
How about other questions? You can even go all wild and turn on your mic and talk to Paul if you want. Paul, did you want to comment on MERS? It's my feeling that governments around the world, we don't really do politics, but governments around the world were, I think, mildly complacent by MERS and mildly complacent, although the Far Eastern countries were less complacent about SARS-CoV-1. And MERS was a coronavirus that appeared and kind of seemed to go nowhere. And I sort of wonder really if European governments weren't fooled by all of that. Even on a kind of scientific level, can you comment about MERS versus SARS-CoV-2?
Yes, I mean, so for that specific virus, it's different. If you imagine a cloud of all of the different coronaviruses that exist in humans and animals and other species, humans and other animal species, it's another example of something that's jumped. And this one jumped from camels into humans. But for some reason, and probably to do with the way it gets into cells, it doesn't use the same receptor to get into cells, and it's not quite such a good virus for transmitting around. It's not so well adapted, and in a way, it's it's quite hard. There becomes a barrier where it's very hard to jump over to become a very well adapted virus and to become something like a SARS coronavirus #2. But there's a lot of us, there's a lot of animals, and there's a lot of viruses. And so these things happen quite often. And it's quite sobering to people, I think, that work in this area, that if you just go back to 2009, so what are we, 12 years on, we've had pandemic H1N1 flu, which we pretty much dodged there because it wasn't a very pathogenic virus, but it went around the world in 2 years. That was followed by MERS. That was followed by the first Ebola outbreak in West Africa. That was followed by Zika virus, which is an insect-transmitted virus originating in Africa, most likely, and then around the world. Then we've had SARS coronavirus 2. So we're on our 5th flurry of things moving into humans over a 12-year period. And at that rate, we are going to be unlucky every so often. And it's hard to imagine it's going to be another 100 years before we have to deal with this again. We did write a number of articles 5-6 years ago saying that this was going to happen, that the best thing the world could do was to get surveillance set up, and that means proper surveillance throughout the world, including in resource poor settings, and that we should monitor by genetics and sequencing. And we wrote an article and we estimated it would cost about £5 billion a year to set up and run that network. And of course, you can imagine what happens when that lands on anybody's desk. £5 billion, my goodness, we can do so much more with that. Well, it turns out that would have been a good investment, I think. But we'll see as we move forward. I think we need to do far more in this preparedness, and it's not just viruses, it's antimicrobial resistance. And I think this is a bit of a wake-up call.
I think, Paul, thank you very much for that. I think on that note, I think we'll formally draw the proceedings to a close. Thank you. Thank all of you for attending, participating, sending in your questions. And once again, Paul, thank you for a great talk.
I'm thrilled, and it's been a great pleasure, and thank you for